Good afternoon. Welcome back to Zoology second year class. We are in the topic, the applied biology SAQs, and some SAQs already we discussed. Next coming SAQs are define the vaccine and discuss about the steps of the types of the vaccines. Here, the vaccines is mainly they are the biological preparations. Here, they are prepared by the same type of microorganisms. These are the vaccines are the biological preparations. They are what is the use of that? They improve the immunity of the particular diseases. For the particular diseases, the for the giving of the immunity purpose, the vaccines are useful. They are made up of they are the biological preparations, and the word the vaccine is coined by the Edward Jenner. Here, next it is containing the what containing in this means the weak microbes are killed microbes are killed organisms are they are releasing toxins are present and also proteins. Here we are prepared the vaccines from the whole organism, the weakened organism, or the with the killed organisms, or the through the toxins and the proteins we can prepare the vaccines. Here the next the biotechnically produced vaccine means we are synthesize the which vaccines they are by the technologically produced vaccines are mainly the first type is called as yesterday only we discussed. One more time I will explain. The last one I didn't, that's why I am taking this one. Attenuated whole agent vaccines. Here attenuated means the disabled or the weakened microbes we are using. Weakened live. Here weak and live microbes are using for the preparation of the vaccines that we call as attenuated whole agent vaccines. Here next these are the mostly antiviral. Here which vaccines the attenuated means the weak and the weak microbes are with disabled microbes for the preparing the vaccines are mainly the yellow fever. For the yellow fever we are taking the vaccines that is prepared by the disabled or weak microbes. Here next the measles, rubella, mumps, and some bacterial with diseases like typhoid. These are prepared by the disabled microbes. Here next is the inactivated whole agent vaccines. Here attenuated means the weak, they are live only, live microbes only. Disabled we can use for the preparation of the attenuated whole agent vaccines. But here the inactivated whole agent vaccines means the killed microbes we are using in this one. They contain killed whole microbes. Example is the influenza. Here for the immunity given taken for the influenza, we are taking this type of vaccines. The cholera and bubonic plague, polio, hepatitis A, rabies, and the polio and the oral polio also prepared by the inactivated the whole agent vaccines. Clear? This one is second type. And the last type, which is the toxoids, means through the toxins, the microbes are releasing the toxins now. Through the toxins, we are preparing the vaccines that we call as the toxoids. Here, they contain the exotoxins. Exotoxins means they release out. That's why we call it the exotoxins. The of certain microbes. There, which one? Diphtheria, tetanus vaccines. Diphtheria and tetanus vaccines are prepared through the toxins the released by the microbes. Here, next the vaccines are injected into the body, and they the immune system react and the products of the antibodies. Here we are injecting that one automatically. Those are taken the vaccine in that people body. The antibodies are produced by memory cells. Already memory cells are present. Through the memory cells, they are releasing of the antibodies. Clear? Yeah. Next, this one is about the toxoids. Clear? Next, whenever the microbes are enter in our body, whenever the microbes are enter in our body, the virulent form, both immediately they kill them and help in the antibodies. Clear? Next, the vaccines prepared in the body. Vaccines, what will they prepare? They prepare the antibodies for the fighting of the diseases. First, fight against the diseases. Clear? This one is about the vaccines, the definition of the vaccine and the types of vaccines. Clear? Next. Next question. Gene therapy. For the improving of the diseases, genetical disorders, the treatment we are taken that we call as the genes through to the genes we are giving the treatment that we call as the gene therapy for the treatment for the diseases. Clear? The gene therapy. What it is? The main is gene therapy. Clear? Gene therapy is the insertion of genes. We are inserting the genes into the cells or tissues. Here, we are inserting the genes. Gene means the chromosome is having the genetic material that we call as the genes. These genes inserting into the cells as well as tissue for which purpose? The treating of the diseases. For the treating of the diseases, we are inserting the genes into the tissues or cells is called as a gene therapy. Gene therapy, gene treatment. Genes are inserted to the cells or tissues for the treatment of the disease. The gene therapy is an anticipated stage and the operation much complicated. Here it is useful, but it is more complicated. Here, next is the basic technique. What technique we are using means a normal gene is introduced into the genome to supplement. Already we are having the genome to that we are giving the normal gene. A normal gene is introduced into the genome to the supplement the abnormal gene. Here, any disease causing gene is in for that disease causing gene, we are giving the supplement normal gene. We are giving as a supplement the vector transfer the therapeutical gene means we are taking the genes from in the vector through the vectors only we are transferring into that particular target cell only. Clear? Next is the gene therapies are mainly the two types are there. What are they means? First is the germline gene therapy. Gene therapy means we are insertion of the gene for the treatment of the disease is called as a gene therapy. Here through the how we are introduced through the vectors. Vectors are transfer the genes to the particular target cell. Next, gene therapy is two types are there. They are called as a germline gene therapy. See this first. Two types of gene therapies are there. First one is called as a germline. Gems. We are introduced into the gems. That's why we call it a germline gene therapy. Germline gene therapy means we are inserting the genes to the gems. Gems means what is the gems? They are the ova or the sperm is called as a gems. Here, next second is called as a somatic line gene therapy. Somatic line gene therapy. Here, for example, we are insertion of the gene through the vector, insertion of gene to the germ cells, either ova or the inside of the sperm. In the fetal stage only, we can do this one. See this one next. The germline gene therapy. In this type of gene therapy, functional gene means which one is we are introducing inside the functional gene genes are introduced into the sperm or ova and integrated into the DNA, integrated into the inside of the sperm and ova. The DNA is there now. To the DNA, we are adding the functional genes that we call as a germline gene therapy. This stage is heritable, means here in the sperms and the ova, means through into the germs, means into the ova or sperm, we are adding or we are giving the functional gene. That means instead of the abnormal gene, instead of that we are giving the functional gene to the ova or the sperm. That is heritable, means the next generations also the disease will be not occur. For the improving purpose, we are giving the functional genes. Giving the functional genes, the abnormal gene is detected and the functional gene is expressing. That is continued in the generations that we call as a that change. This change is inheritable, but it is not easy, means it is a complicated one. And also this therapy is still in the infant stage only. In the infant stage only, this therapy will do. After in the adult stage, we can't do this type of gene therapy. Clear? The germline gene therapy means we are in insertion of the functional genes to the abnormal genes. Clear? Any abnormality, for example, the color blindness is there, and also some what it is hemophilia are also called as the muscular dystrophy and the phenylketonuria. Like that, number of genetical disorders are there. That genetical disorders are mainly we are treated by like this. Clear? Germline gene therapy. Through the germs, we are giving the adding the or insert the gene is called as the germline gene therapy. Next is called as a somatic line gene therapy. Somatic cells. Here we are giving the germ cells first time. Here the germline gene therapy means we are giving to the germ cells. We are the genes are introducing. But here in the somatic line therapy, what it is means in this time, normal gene is inserted to the somatic cells of the patient. Those are having the any disease. For the recover or treatment of that disease, we are inserting the gene into the somatic cells. What are the somatic cells? Here, we are having the means the sex chromosomes and the autosomes are there. Like that only here also germ cells are there, somatic cells are there. The germ cells are there for the determination for the purpose they are useful. Here, somatic cells means other than the germ cells is called as the somatic cells, and the change that takes place temporary, not heritable. Before that, germline gene therapy means they are in that generation, the change will be there, means the disease will be disappear, and that's also next generation that is not coming. That is the germline gene therapy. But here we are introduced the genes into the somatic cells, that is temporary and not heritable. It is in that temporary means in the lifetime also total not occur, but they are sometimes in that lifetime only they will reoccur the disease. But in the germline, this is a permanent and inheritable. Here this one is a temporary and not inheritable. Here next, the insertion of the gene may take place either outside of the body. Outside of the body, we are giving the means in the test in the lab. In the lab, we are giving the genes to the somatic cells that we call as a what it is. That is called as a ex vivo. Here, ex vivo means we are taking the outside insertion of gene. Insertion of gene to somatic cell. Somatic cell. Here, outside insertion is called as a ex vivo. Here, next in vivo means the inside of the body. Inside of the body, insertion. Inside of body, insertion of genes. Insertion of genes is called as, to the somatic cells is called as a in vivo. See this one here, there. ex vivo. The R inside of the body is called as a in vivo. First, the first clinical gene therapy was given to 4 years girl. First gene therapy after the experiment, first gene therapy is given to the 4 years girls in the 1990, treated the disease that is called as a adenosine deaminase deficiency, ADA, means adenosine deaminase deficiency. The treatment purpose, they are using this type of gene therapy. Which gene therapy? Somatic line gene therapy. Here, next is this disease, this deficiency caused the serious combined SCID, means the serious combined immunodeficiency is caused. Which one? The deficiency of this one. Here, this substance deficiency, the means the adenosine deaminase deficiency causes the what it is? Severe combined immunodeficiency, means the several immunological disorders are occurred that we call as a serious combined the immunodeficiency. Here, next the deficiency may be cured by bone marrow transplantation. Here, this disease is mainly cured by the bone marrow. This deficiency may be cured by bone marrow transplantation. Bone marrow transplantation also we can prevent this type of disease, or by the enzyme replacement therapy. Also, we are by the enzyme replace therapy also we can cure this one. Clear? Enzyme replacement therapy. Clear? In either case, it is not a completely cure. Here, we can cure the adenosine deaminase deficiency with the bone marrow transplantation or also enzyme replacement therapy. Two ways we can cure this disease, but it is not a complete cure. It's sometimes it will maybe occur next also. That's why it is not a permanent. Clear? It is a temporary. Next, the genetically, see, observe here. Genetically modified DNA can be inserted to the cell transcription or by the gene guns. Clear? How we are inserting the generally modified DNA, or DNA means genes, inserted to the cells by the transfection or by the gene guns. They are in the laboratories. They are using these things, and the synthetic here next the synthetic the oligodeoxynucleosides. Clear? Synthetic oligodeoxynucleosides are used for the inactive the disease causing genes. Here for the genes, which genes are causing the disease, that genes are inactivated by the synthetic oligodeoxynucleosidase. Here, this is about the gene therapy. The gene therapy means the insertion of the genes into the target cell or the abnormal cell, that we call it as a gene therapy. These two ways are there. One is called as the germline gene therapy, and second one is called as a somatic gene therapy. To the germs, we are insertion of the gene. We are inserting the gene to the sperm or ova. That is called as a germline gene therapy. It is a heritable, means it is transferred to the next generation also. But it is not easy. It is not easy as said. Here, this therapy is still in the infant stage. In the adult stage, it is not effective. That's why in the infant stage only they can do the germline gene therapy. Here, next, somatic line gene therapy means the genes are insertion to the somatic cells, that we call as a somatic line gene therapy. It is a temporary. This one is a permanent, but it is not easy. Here, permanent, and they are heritable to the next generations also. But in the somatic line therapy, it is a temporary and non-heritable. Here, and it is mainly it is two ways we can give. Here, in the outside the body we can give, and also inside the body we can give. That we call as a ex vivo or in vivo. Clear? This one is about the gene therapy. Gene therapy using for the curing of the abnormal genes, or curing of the prevent of the genes through the insertion of gene. Clear? This next. We are going to the next SAQ. Next, the features of silent features of the cancer cell. This is not that much important, but it is sometimes it also will ask. That's why I'm taking this one also. Means the cancer, the four silent features of the cancer cells. What are the features of the cancer cells? We are going to discuss. The cancer is a syndrome characterized by the uncontrolled, unrestrictable, the purposeless cell division. Here, normally cell division is a restricted, their cell division is occur. Here, regular manner they will occur, controlled they occur. But here, some organs inside of the some organs are some places the cell division is uncontrolled and the unrestricted and the purposeless cell division is occur that is causing the tumors that is called as a cancer. Cancer is a syndrome characterized by the uncontrolled or unrestricted purposeless cell division and more cell division. For example, the 10 cells are forming. Instead of 10 cells, here the 1,000 cells are forming after the cell division. That causes the tumors that is called as the cancer. Here, next is due to the failure of the cell cycle regulation. Cell cycle regulation is not there automatically. The cell division is more. It is uncontrolled and unrestricted and purposeless. Here, next is what are the silent features of the cancer? What is cancer? We discussed. Cancer means the cells are more divided. It means the uncontrolled and the unrestricted purposeless cell division is occur that causes the tumors that we call as the cancers. Here, next is what are the features? Means lack of contact inhibition. Contact inhibition is lack. What it is means the normal cells stop cell division and cell mobility when they are close contact. Here, there some normal cells are stopped to the cell division and the cell mobility. They are closely contact. That time what will happen? Cell division is not occur. But this is called it as a contact inhibition. Cancer cells not like this. Here, for example, this one is a one cell and this one is a one cell. The attachment will be there between the cells. The attachment is there. The attachment is tight, and what will happen means there is no chance to division, cell division. That is called as a inhibition. Here, contact inhibition. Contact inhibition means they are arranged the cells. Here, that time what will happen means no chance to the cell division. That one is lost. The contact inhibition is lost. That's why the cells are undergoes the division and form it as a tumor. See this one. Cancer cells lack this property, hence they are continuous to grow and divide. Here, the space is there between the two are contactly. Their cells are arranged compactly. What will happen? No chance to divide. If that is loose in the cancer cells, what will happen means easily they can divide and make it as a number of cells and form it as a tumors. Here, next this is the one property, lack of contact inhibition. Contact inhibition means closeness of the cells. Here, next is the lack of cadherins. Here, what is the cadherins means? The cadherins are absent in the cancer cells. Hence they are detached with the cause the metastasis. Means the cadherins means thread-like structures are attached between the cells. That cadherins are absent in the cancer cells. Cancer cells are not having the thread-like attachments. That's why they are detached and they are detached to the other cells and they are causing the metastasis. Means moving from one place to another place that we call as a metastasis. Yeah, that is cadherins means they are mainly the thread-like structures between the cells. Yeah. Next is the immortalization. Mortality means death. Immortalization means if the DNA is damaged in normal cells, they are repaired and damaged in the time. Here, if the DNA is damaged inside of the cell, normal cell, what will happen? They can repair the damage in time. Here, if they fail to repair and damage the DNA, they undergoes the programmed cell death. Here, if the DNA is not the repair, what will happen means they will be die. But through the apoptosis. Means through the apoptosis they can die. Means the DNA is damaged automatically. The normal cell will be damaged. If we not repair, they damage, and that we call as the apoptosis. But cancer cells do not undergo apoptosis. For example, see this one. Here the normal cells is there. Now inside of the normal cell, if the DNA is there, if the DNA is damaged, automatically they will be, means they will repair. They can, okay. Otherwise, they can damage, means they can destroy, say apoptosis. Through the apoptosis, means the cell will be cause death. That is the normal cells. But in the cancer cells, if the DNA is damaged, that time also this cell will be not damaged. That is the immortalization. They are not undergoes death. The cell is not undergoes death. That we call as a immortalization. Here, next is the but cancer cells do not have this one. Next is the angiogenesis. Angiogenesis. In fast-growing cancer tumors, in order to get more oxygen and nutrients, the tumor attract more blood vessels. Here, the tumors are there, na? Tumors, other than the normal cells, the tumor means the cancer cells are there, na? They are mainly the attract to the blood vessel from their surrounding of the matrix. They are angiogenesis, means the more attracted, the blood vessels are more enter inside of the cancer cells only, and the blood supply is more for that we call as a angiogenesis. Means the fast growing cancer tumors, they can get the more blood circulation. Why? Means they are, in order to get the more oxygen and nutrients, they blood supply is also more. That condition is called as a angiogenesis. Means how it is occur means cancer cells mainly having some features are there. The 4 features are there. We will discuss already. The lack of, first one is the lack of the contact inhibition, lack of cadherins, and lack of immortalization. Cadherins means thread-like structures between the cells. Immortalization means normal cells, the DNA is damaged, that automatically die, but cancer cells are not die. That we call as a immortalization. And angiogenesis means the tumor cells, fastly increasing the tumor cells are getting the more blood supply and the more oxygen and more nutrients. That we call it as a angiogenesis. Here, this one is about the features of the cancer cells. Next is the types of cancers. Next one is the types of cancers. Here, the names means first one is the based on the origin. Cancers are origin where they are present. Where they are origin. With that, we are divide the cancers into these types. First one is called as a carcinomas. Here, the name only carcinomas means the cancers are cancers of epithelial or epithelial tissue or cells, epithelial cells or epithelial tissue, that we call as a carcinomas. It is the most common type of cancers where the epithelial cell is present, epithelial tissue where they are present. Every time covering of the body, covering of the total organism and total all the organs in our body. How many organs are there? All the organs outer lining is lined by epithelial tissue only. Means any outer line, the tumors are formed. That is which type? That is called as a carcinomas. Here, epithelial cells are the epithelial tissues are having the tumors or the cell division is more. That type of cancers we call as a carcinomas. it may be affected mainly the epithelial tissue or epithelial cell cancers. Here, it is the most common type of cancers. Here, next second type is called as a carcinomas. Sarcomas. Carcinomas over na sarcomas. Sarcomas means connective tissue, means inside of the body. Now, this is the epithelial tissue is the outer covering. The inside of the organs, the connective tissue means mainly connective tissue binding the organs, organ systems, and all the tissues. That connective tissue is in the connective tissue. Any tumors are formed or cell division is more. That type of cancers is called as a sarcomas. First one is affected the epithelial tissue outer lining, that we call as a carcinomas. Sarcomas means the effect the cancers are appear in the connective tissue. Yeah. Next one, third one is called as a leukemia. Means this one is a liquid tumor, blood cancer. We are saying that only. Here a liquid tumor. It is a cancer of bone marrow resulting the unrestricted production of the WBC. Bone marrow. What is a bone marrow? Bone marrow is actually erythropoietic, not hemopoietic organ. Means what is a hemopoietic organ? Bone marrow. Hemopoietic organ. Means why we call it as a hemopoietic organ means that is producing the RBC, all the blood cells, RBC, WBC, and also thrombocytes are also called as a blood platelets. Blood platelets. All are synthesized by the bone marrow. That's why we call it as a hemopoietic organ. Here, what is it? Leukemia. Leukemia means it is the liquid tumor, means the inside of the blood it is appear. Liquid tumor, it is a cancer of bone marrow. In the bone marrow, any tumors are occur automatically, resulting unrestricted production of the WBC. WBC is more producing, the more than the average abnormal increase is there, that we call as a leukemia or the white blood cancer. This is also called as a leukemia. Next, this is about the leukemia or liquid cancer. Next one is called as a lymphoma, lymphatic tissue. Here, like a blood circulatory system, the lymphatic system is also there. Lymphatic system is having the lymph capillaries, the lymph vessels, lymph nodes are there. Tumors are occur in the lymph capillaries, lymph vessels, lymph nodes, or lymph glands also. They are affected, that we call as a lymphomas. Here, the cancers of the lymphatic system is called as a lymphomas. Here, next is the familiar cancers. Here, with the family, the cancers are occur, that we call as a familiar cancers. Here, that the cancers that are inherited from the parents and the grandparents. Inheritedly, they are occur. For example, if the grandparents are having next generation, they can get, that is called as a familial cancers. Next, the sporadic, without the family history. Family history, nobody is having the cancer, but the people are getting that type of is called as a sporadic cancers. Here, cancers that occur without any family history, that we call as the sporadic cancers. Here, these are about the types of cancers. Simply, the 4 types, so you know easy way I'm saying, see here. See this one, types of cancers means first one is called as a carcinomas. Carcinomas. Carcinomas means just the cancers of epithelial tissue. It is a common. Here, carcinomas are mainly the causing in the epithelial tissue or cells. Epithelial tissue or cells. That type is called as the first one. Simple one, the carcinomas, that is the cancers of epithelial tissue. Here, next, that is a common type of, mostly it is a common and curable cancers. These are here. Next, second type is called as a sarcomas. Sarcomas. Which one? Sarcomas means which tissue is affected? Connective tissue. Here in the connective tissue, the cell division is more and form it as a tumor. That type of cancers we call as a sarcomas. Here, next third type is called as the leukemias. Leukocytes. WBC is called leukocytes now. That's why here it is a leukemias. Leukemias. Here, leukemia means the leukocytes are more. Leukocytes means WBC. The inside of the bone marrow, cancer is occur means the cell division is occur automatically. This is causing the production of the WBC is more. This is also called as a leukemia. Is also called as a inside of the blood. It is there. That's why we call it as a liquid tumor. Liquid tumor. Next, fourth one is called as a lymphomas. In the name only there. Lymphomas means the cancers of the lymphatic system is called as a lymphomas. This is depending upon the region, depending upon the origin. These four types are there. Here, what it is? Carcinomas, sarcomas, leukemias, and lymphomas. Last two is there. What are they? See here, this one. Here, these two are there. Familial cancers and the sporadic cancers. Familial cancers means cancers are occur that are inherited from the parents to grandparents, inherited to one generation to next generation. With the inside of the family, the cancers are occur that we call as a familial cancers. Next is the sporadic means it is not depending upon the family, not inherited. That's we call as a without any family history. The cancers are occur suddenly. The cancers are occur. That type of cancers we call as a sporadic cancers. There. Next. Next one is a MRI. The magnetic resonance image. These are the mainly for using purposes, but the diagnose the diseases purpose. We are using the some devices that we call as a MRI. This is called as a the stands for what it is. What is the full form of MRI? I'm writing here. See this one: magnetic resonance image. Magnetic resonance image. The full form of MRI is the magnetic resonance image. And what it is means, this one is mainly magnetic. In this we are using the magnetic tube we are using. Here it is a radiological technique. Here diagnostic radiology technique. Here next is the MRI does not use the ionized radiation as X-rays. The X-rays are having the more ionization radiation is present, but in the MRI magnetic resonance image, that much ionization is not there. Here next that's why it is a safe procedure. Why the comparatively MRI X-ray, which one is better means MRI only. Why means MRI is not having the ionizing radiation. That's why it is the safest procedure. Here next it produces the detailed picture of the organs. For example, any inside of the organs or tissues or bones, any structure we want, detailedly we want, that time what it is, the procedure, which procedure we are using? MRI means we went to the physician, any defect is there, inside of the bone, any defect is there, inside of the organs, any problem is there. That time what they suggest means for the MRI, means they to keep the for a particular organ or particular bones, he suggests that they take the MRI. Here next the procedure detailed picture of the organs we get, and the soft tissues and the bones pictures we can get, and other structures of the body. Any other structure of the body, organs, bone structures are we are getting in this technique. Here Next, the MRI scanner is how it is there. All of you, all you know, in the hospitals, they are round. The tube-like structure is present. That we call as a circular magnetic tube. Gaint means large. They are here. MRI scanner is a gaint circular magnetic tube. First is this one is visible here. Next is the patient is placed into the movable bed. The surrounding round, sorry, for example, like this. Like this, the magnetic tube is present. The magnetic tube, this one is actually magnetic resonance. The tube is there. Here the movable bed is there. Here the same. Roughly, I'm drawing the diagram for understanding purpose. Here the patient is placed. Here, see this one. The patient is placed in the movable bed that moves into the magnetic. The push. This one is pushed inside of the. This one is movable magnetic tube. Movable magnetic. Inside of the tube, movable magnetic tube, the bed is there, movable bed is there, that is pushed inside of the not movable, it is the movable bed is there. Sorry, movable bed is there. Inside of the bed, they are inserting the patient. Here, this is it. The patient is placed in a movable bed that moves into the magnetic ring. Here, next is now what they did means human body is mainly composed of water. 92%, 99% of water is present inside of the body that which is contain the protons. Here H2O, number of protons are present. That is the magnet create the strong magnetic field. This magnetic tube is creating the magnetic field. When the patient is inserted to inside, what will happen means the human body is having the more amount of water, means the 99% of water is there. With that water, the inside of the water, protons are there, number of H ions are there. That is called as a protons. The magnet create the strong magnetic field. It create the strong magnetic field. The body protons alignment will be there. Here, if the magnetic field is increased, the magnetic field is appeared. That's time the body alignment will be formed. Here, create the strong body, the proton alignment with the direction of the magnetic field. Next, the first radiation time, what will happen? The patient total alignment will be appear. After that, second radio frequency electromagnetic field turned on. Here, now electromagnetic field. First, before that, the magnetic field they are applying. That time, body alignment will be occur. First, body alignment will occur. Second, after the alignment is over, the second radio frequency electromagnetic field is turn on in the brief. Some period only they are applying the radio magnetic field. Here, next, what will happen next? The protons of different parts of the body release different energy. The time electromagnetic field is applying, what will happen means body water is having the protons now. That protons are releasing different wavelengths of different waves of the energy is releasing. Here, see this one. The protons, different parts of the body release different energy. Different energy they are releasing means the color of the waves also different. Here, which detected by the MRI scanner. Here, that they are releasing the energy now with that releasing energy. Which one is that detected by the MRI scanners are detecting that one. Here, accordingly, there is a contrast between the images, the different tissues based on the water content. Here, for example, bones are having the less water content. Muscles are having the more water content. That's why the bones are appear in the white in color. Here, like this, depending upon the quantity of the water, different types of colors will be formed. See this like this. Accordingly, there is a contrast between the images of different tissues based on their water content. Water content, depending upon the water content, they are different structures they will give. Here, next, the information received is processed by the computer. The total is processed by the computer, and the image is generated. Here, first wavelength, first magnetic field is applying by the magnetic tube. After that, the brief period, electromagnetic frequency, the field will be on. Automatically, what will happen here? The protons are there inside of the body. Protons are they are releasing the different types of energy. With that, they are forming the picture is formed, the image is formed, that is taken by the MRI scanner. MRI scanner is detected this proton energies. Here, next, according to the contrast between the different tissues based on the water content. Here, next, that is given to the computer, and the image is generated. Here, this image is we are getting the image now. Image is transferred to the photographic film. Here, we are taking as the image photographic film. Sometimes radio contrast agents like gadolinium is used for increasing of the accuracy. For that clear picture, that time they are using the gadolinium. They are using for the clear picture. Here, this is about the MRI. See, on one more time, I will explain the MRI. Simply briefing by MRI. And here in this lesson, the left, the two SAQs are there, more two SAQs are there. That is one is the ECG, and another one is the ELISA, enzyme-linked immunosorbent assay. With these two, we completed the SAQs. Once I am briefing this one, and listen here. Here, the MRI. Here, what is the full form of MRI? That's a magnetic resonance image. All of you saw this one, and the circular magnetic field tube is present in this one. It is actually it is a diagnostic radiology technique. Means in the diagnostic only we are, means in the hospitals also it's there. It is a diagnostic radiology technique. It does not having the ionization radiation like a X-rays. Like X-rays, it doesn't have the ionizing radiation. Here, that's why it is called as the safest processor. Two marks, they will ask why the MRI is safe. The MRI is safer than the X-rays. This one only. Here, MRI doesn't have the ionization radiation. That's why it is the safest procedure. Next, it produces the detailed picture of organs. MRI, the X-ray is giving the picture for the more bones, but it is not total clear picture. They are giving more than the X-ray. This one is giving the clear picture of the organs, soft tissues, bones, and other such structures of the body. Any structure, any organ, clear picture. They want to see physicians. That time they suggest the MRI. Here, the MRI scanner is a main apparatus. Is mainly they are having the gained circular tube is biggest gained circular magnetic tube is present. The patient is placed in the movable bed. Through the movable bed, they inserted inside in the magnetic ring. After the magnetic ring, what will happen means they producing the ionizing energy. That time, the human body is having the mainly more amount of water is present, which contain the protons. Here, already I said now, the magnetic create the strong magnetic field. The body proton alignment will occur. First magnetic field time, the total body alignment will be formed. Next, second radio frequency electromagnetic time. They are brief time. Depending upon the releasing of the energy. The protons already taking the magnetic field. They are releasing of the electromagnetic field. We are applying that time. They are liberating the different types of energies. That time they are form it as a different structure, different form that is detected by MRI scanner. Here, next, it is accordingly these contrasts between the images of the different types of tissue depending upon the water content. Clear? Next, This is taken by this magnetic field. After that, they taken into they processed by the computer, and the form it as the image is generated. That image is transferred it as the photographic film. Here, next this one is some clearance purpose. The MRI, we are using the gadolinium. Here, next this one is about the MRI procedure. Here. See this one, I will show which one we discussed in the last lesson. See here. First MOET, the multiple ovulation and the embryo transfer method. This is the artificial method we are using. We are discussing this one, and next is avian flu. Here, this one is the second question. Avian flu. This is caused by the H5N1. Here. It is caused by the H5N1 virus, the flu-like virus, and it is a pandemic disease. It is not only it is occur in the human being also. It is a pandemic disease or epidemic disease, and how it is mode of infection is by touching of the contaminated surfaces and the bird releasing. Which one? Through the saliva and through the fecus. Here. Next, what are the symptoms in the human means? They are having the typical flu-like symptoms, fever and the dry cough and the breathing difficulty and the headache like this. The symptoms will occur in the avian flu. Next, the economic importance of honeybee. Queen bee also we discussed. And the next is the economic importance of honeybee. Honeybee is mainly the like a honey. As a honey, and that is the nutritious food that is having the sources of the fructose, glucose, vitamins and minerals they are having. And the second one is called as the beeswax, that is useful for the preparation of the cosmetics, polishes and candles. And one more, propolis is there, that is useful for the treatment of the burns and wounds. And the bee venom is there, that is useful for the treatment of rheumatoid arthritis. Clear? Next is the bees are the pollinators for the pollinating of the plants. Next, the insulin structure we discussed. In the insulin, mainly the polypeptide chains are there. The two main polypeptide chains are there. They are having the, how many amino acids are there? 51 amino acids are there. One is having the 21 amino acids. Another chain is having the 30 amino acids. Both the chains are connected by the stretch. That is called as a C polypeptide. Here, the two chains are held together by the disulfide. Between the two chains, the disulfide bonds are there. And the insulin is primarily synthesized by the prohormone. Here, a prohormone that is called as the proinsulin. From the proinsulin, the insulin is formed. Here, which one, how it is, the stretch will be released, the insulin is activated. Here. Next, today only we discuss about the types of vaccines. The vaccines are 3 types are there: attenuated and the inactive whole agent and toxoids. Next is the gene therapy. For the preventing of the diseases, the insertion of the gene, that we call as a gene therapy. The gene therapy is the 2 types are there. They are called as germline gene therapy and the somatic line gene therapy. We inserted the genes to the germ cells and like ova and sperm, that we call as a germline gene therapy. We are introduced the genes into the somatic cells, that we call as a somatic line gene therapy. Clear? Next, the cancer cells features we discussed. That is not that much important. Next one is the cancers. 6 types of cancers are there: carcinomas, sarcomas, and the leukemias, and the lymphomas, and the familiar cancers and the sporadic cancers. Yeah. Next, we discuss about the MRI. Here, now only we discuss about this magnetic resonance image for the images of the organs, the soft tissues and bones. In this, mainly magnetic field we are applying. 2 magnetic fields we are applying. First one is the normal magnetic field. Second one is called as a electromagnetic. See this one, frequency of the electromagnetic field. We turn on brief time. That time the picture, the body, organs, and the bones pictures will come. That is about the MRI, magnetic resonance image. The 2 SAQs are left. They are the ELISA and the ECG. Next class, tomorrow's class, we will discuss in the next class SAQs, and we start the VSAQs. Here, let's have one by one lesson and taking the VSAQs. The first I completed these 2 SAQs. Afterwards, the VSAQs we will discuss. Okay, tomorrow same time we meet this in this class.